Inside every human cell is a special compartment called the nucleus, which contains chromosomes that store our DNA, kind of like a library, with all of the genetic instructions on how to build our bodies. In order to send these instructions across the body, DNA needs a messenger. This is called RNA. It's a photocopy of a particular gene from the DNA that carries all of the specific information on how to build a protein. RNA is made up of two major components: exons, which contain readable genetic code, and introns, which don't code and can't be read, but still contain important information. It's just like building a recipe. To turn the RNA into a continuous, readable message, the introns must be cut out and the exons must be pasted together. This is called RNA splicing. But it doesn't just happen on its own. The thing responsible for cutting and pasting the RNA message is called the spliceosome, which is made up of many different parts that each have their own job. During RNA splicing, they all work together to complete 4 steps. They find the specific instructions that mark the beginning and the end of an intron. They bring the 2 exons close together. They cut out the intron at each end. And they paste the exons together. When someone is born with uncommon patterns in their genes, it can confuse the spliceosome, which means they might make mistakes when cutting and pasting the introns and exons. When this happens, it changes the way messenger RNA looks, which means it can't be read and understood. When the instructions can't be read properly, the body can't build itself properly. This explains why some people have genetic diseases and neurological conditions, because variants in their genetic code can disrupt their gene splicing.